Red Lano, Red Latinoamericana de Neuro-Oncología.
Let me start by thanking the organizers for inviting me to Red Lano and to beautiful Cartagena in Colombia. It's my first trip to Colombia. Enjoyed it very much. So I'm going to talk to you this afternoon about anti-angiogenic therapies for glioblastoma. I will focus on bevacizumab, but we'll cover a couple of different agents that are currently under study for glioblastoma. First, I thought it would be worthwhile to review some of the background of cancer-related angiogenesis. There are multiple proangiogenic mechanisms of action involved. Perhaps the classic notion is that once a tumor reaches a certain critical size, it begins to elaborate a number of cytokines that can have both local and remote effects. Some of these local effects involve cytokines that induce sprouting from normal adjacent blood vessels, so ingrowth of blood vessels into the tumor. Some of the remote effects of these cytokines is mobilization of endothelial cell and other progenitor cells from the bone marrow, which in turn migrate to the site of the tumor and are incorporated into the tumor vascular network. At some point, it appears that subpopulations of tumor cells become independent of these new blood vessels and can actually co-opt existing native blood vessels and grow along normal blood vessels within the brain. This is so-called vessel co-option. So there are multiple mechanisms of action here, which is important when you think about this therapeutically. Moreover, there are multiple proangiogenic signal transduction pathways. To date, we have focused on this one, the vascular endothelial growth factor, ligand, and its receptors, and the downstream effects of ligand binding to receptors to promote endothelial cell proliferation, survival, migration, and promote angiogenesis. So our therapeutics have really focused on blocking the ligand, blocking the receptor, or even blocking some of the downstream signal transduction, part of the downstream signal transduction pathway. But it's very clear that there are other proangiogenic signal transduction pathways, multiple, and ultimately effective anti-angiogenic therapies will need to address some of these other pathways. I will touch on one or two of these today, but in the interest of time, it will be brief, but you can see just some of the other pathways here, the angiopoietin 1-2, Tie2 receptor signal transduction axis. There are integrins, there are fibroblast growth factors, there are many other pathways involved. We also know that glioblastoma, on the surface at least, is an attractive target for an anti-angiogenic approach. In fact, microvascular proliferation is one of the pathological hallmarks and requirements to make the diagnosis of glioblastoma under the microscope. Much of this is driven by hypoxia within the tumor that leads through a series of intermediate signals to ultimately endothelial cell division, proliferation, survival, migration, and angiogenesis. It's known that the VEGF ligand and receptor levels also correlate with the grade of glioma. Thus, the higher the level of the VEGF ligand or receptor, the higher the grade of glioma. So at first blush, it appears that this would be a tumor like renal cell carcinoma, thyroid carcinoma, that would be a very attractive candidate for drugs that target angiogenesis. We're not quite sure how these drugs actually achieve a therapeutic benefit, not only in glioblastoma, but in other solid tumors. And there are a number of hypotheses that are out there. And these, by the way, are not mutually exclusive. And different pathways or different mechanisms could be active at different times during the growth of a tumor. The classic notion put forth in the 1970s in a famous paper in the New England Journal by Judah Folkman is that of neoangiogenesis, or the ability of these drugs to actually directly target endothelial cells, leading to endothelial cell death, vessel pruning, and so-called starvation of the tumor and tumor cell death. Another hypothesis is vessel normalization, and this is the work really of Rakesh Jain, who's at my institution, and the idea here is that these drugs actually transiently normalize the vascular network, which improves oxygen content or oxygen delivery to the tumor, mitigates tumor hypoxia. And we all know that in that setting, our cytotoxic therapies like ionizing radiation or chemotherapy work better. Also, it may be that there's better perfusion or blood flow into the tumor during this period of normalization, which may enhance the delivery of chemotherapeutics. And we also know that when the vessels normalize, they become less leaky. So they become less permeable, and therefore there's a beneficial effect during vessel normalization on tumor-associated edema. Another idea in a nice paper a few years ago in Cancer Cell by Richard Gilbertson and his colleagues at St. Jude is the notion that these drugs actually disrupt a very critical perivascular cancer stem cell niche, and that when this niche is disrupted, you can actually see death of some of these glioblastoma stem-like cells. This was very nice preclinical work. And recently there has been the notion that anti-VEGF agents, at least, might, through their vessel normalization, enhance innate anti-tumor immunity and improve trafficking of immune effector cells into the tumor and improve an enhance an anti-tumor immune response. So there are different ideas about how these drugs actually work. The initial report of the benefit of Bevacizumab, which is a humanized monoclonal antibody against the VEGF ligand, was actually by a community oncologist in Texas. And she administered Bevacizumab with a chemotherapeutic, irinotecan or CPT11, and she noticed that several of her patients with glioblastoma had regression of their tumor by MRI. This work was presented years ago at a meeting in Scotland, and there was a race to the exits to see who could get home fastest enough to launch the first clinical trial of Bevacizumab in recurrent glioblastoma. And this was one of the first studies reported, and this was a study out of Duke in which patients with recurrent glioblastoma were administered Bevacizumab in combination with CPT11. And the results were striking. A small phase 2 study, but over half of the patients had objective radiographic responses, regression of contrast-enhancing tumor, like in this MRI here. Here's before and after, same case, a coronal view, before and after. And there's not really any argument that contrast-enhancing component of the lesion is much smaller. Moreover, they showed in this trial that about half of the patients were still receiving the drug without any tumor progression at 6 months. And normally we would expect anywhere from about 15 to 20% of patients receiving chemotherapy, for example, would last 6 months without any tumor progression. So this metric too, was an impressive number. So that led to a randomized trial. Now this was the brain study in recurrent glioblastoma, so all of these patients had failed prior surgery, radiation, and temozolomide. It was a non-comparative randomized phase 2. There was central review of all of the MRI scans to determine response. Primary endpoint was progression-free survival at 6 months, which I just alluded to. 167 patients were randomized. These are the results. The overall radiographic response proportion was impressive, 30 to 40% in both arms. Remember, both arms had Bevacizumab within them. And 40 to 50% of the patients were without any disease progression at 6 months. So the PFS6 also held up in this trial. Data from this study and one other non-randomized phase 2 study that was conducted at the National Cancer Institute was reviewed by the Food and Drug Administration in the United States, and it led to an accelerated approval of Bevacizumab as monotherapy for recurrent glioblastoma. That approval was contingent upon subsequent studies demonstrating a survival advantage of Bevacizumab in this disease, and we'll come back to that. So the drug was approved in 2009. It was rapidly adopted into practice in the United States, both tertiary academic centers as well as community oncology practices. It's a drug that's now widely used for glioblastoma patients. In fact, it's a rare patient with glioblastoma now in the United States who doesn't receive this drug at some point in their disease course. Well, subsequent to that approval, there were a number of other randomized trials, in this case, randomized phase 3 trials that were launched and successfully completed. Let me tell you about a drug we were involved in. This is not Bevacizumab, but this is an anti-VEGF drug. It's an oral anti-VEGF drug, Cediranib, very potent oral inhibitor of VEGFR2, which is a key receptor in cancer-related angiogenesis. It's about 50 to 100 times more potent than sorafenib and sunitinib at that receptor, which are FDA-approved drugs for various indications. Half-life is such that you give the drug once a day. The side effect profile of this class of agents is well known and is listed for you there. Based on phase 1 and promising phase 2 data, we launched a randomized phase 3 trial in recurrent glioblastoma. This was the REGAL trial. This will be published in the Journal of Clinical Oncology within a few weeks. And again, these are patients who have failed surgery, radiation, and temozolomide. And in this trial, they're randomized to receive either Cediranib alone or lomustine, which is an oral alkylating drug, with Cediranib, or lomustine with a placebo. So it's partially blinded. 2 of the arms were blinded. 325 recurrent glioblastoma patients. Our primary endpoint was progression-free survival at 6 months. This is the result. These were all centrally reviewed MRIs, and although there's a trend favoring the Cediranib-containing arms versus the lomustine arm, these did not reach statistical significance. Overall survival, similar story. No significant difference in overall survival in the lomustine arm versus the 2 Cediranib containing arms. So this drug did not meet its endpoint and will not be approved for glioblastoma. Subsequently, these 2 other randomized trials were also launched in newly diagnosed glioblastoma. So the company and with advice from others decided that the randomized trials that would be conducted to get Bevacizumab fully approved would be done in the newly diagnosed glioblastoma setting. And the setup for these 2 trials, one was Avaglio, conducted largely in Europe. The other one was RTOG-0825 in North America, was a similar design. Both were newly diagnosed, would have their surgery, and then they would go on to either Bevacizumab, temozolomide radiation, or a placebo, temozolomide and radiation. So there were some, here are the 2 trials kind of laid out. Avaglio, industry-sponsored. The Avaglio trial, importantly, allowed biopsy patients to enroll, whereas the RTOG trial did not allow biopsy patients to enroll because they were using tissue to stratify for MGMT as well as a genetic panel. In the Avaglio study, there was mandatory participation in all of the quality of life studies that were secondary endpoints in the trial, whereas this was voluntary in RTOG-0825. These were the outcomes. Now, these have not been published, but they have been presented just last month at ASCO in Chicago. This was some of the public data that's presented. In Avaglio, there was no improvement in overall survival in the experimental arm. There was, however, a significant improvement in progression-free survival. You can see a hazard ratio of 0.6 and a highly significant p-value. There was also, in secondary endpoints, there was improvement in quality of life as measured, and there was longer functional independence for the patients who were randomized to receive Bevacizumab. In contrast, in RTOG-0825, similarly, there was no improvement in overall survival, and there was also no significant prolongation of progression-free survival. There was a trend favoring the Bevacizumab arm over the placebo arm, and this p-value, while it may look significant, did not reach their predefined threshold, which was 0.002. So it came very close, but no cigar. The interesting thing is, presented at ASCO, it appears that those patients who received Bevacizumab tended to have more symptoms than those who were not on Bevacizumab, and there was a suggestion, at least on some of the neurocognitive measures, that there may have been some decline in neurocognitive function in the patients who received bevacizumab. So this is a summary of where we stand with, in terms of randomized phase 3 trials in anti-angiogenic therapies in glioblastoma. I will add parenthetically, I won't dwell on it, but parenthetically, this data from the Selingitide trial, this is an anti-integrin drug which might have some anti-angiogenic properties, was presented at ASCO last month. And this was also a study in newly diagnosed glioblastoma, but only in those patients who were MGMT methylated. So they were randomized to a Selingitide arm and a non-Selingitide arm, and there was no improvement in either progression-free or overall survival. So of 4 anti-angiogenic randomized phase 3 trials, none of them have resulted yet in an improvement in progression-free survival. One of them has resulted in improvement in progression-free survival. Another one came very close but did not meet the pre-specified statistical threshold. There were 2 other trials I want to tell you about at ASCO, which were randomized phase 2 trials that also addressed bevacizumab. I will maybe confess my bias here that I think that this design with bevacizumab is probably the design that I would have elected to have in a phase 3 trial, which is to say to study it in recurrent glioblastoma. And this was set up just like the cediranib trial. We have oral lomustine alone versus bevacizumab alone versus bevacizumab plus oral lomustine. And in this randomized phase 2 trial, there was a significant improvement in progression-free survival and overall survival in the bevacizumab CCNU arm. There's also an EORTC randomized phase 2 with a very similar design, but we don't have those results yet. Another, this was the so-called BLOB trial. Another trial that was first reported at ASCO last month with bevacizumab was the GLARIUS trial. And in this trial, European study, they took newly diagnosed glioblastomas, but the poor prognosis subset of those that with unmethylated MGMT. And those patients, after their surgery and after their temozolomide and radiation for 6 weeks, received either more temozolomide or received bevacizumab and CPT11. And they reported a statistically significant progression-free survival advantage for those who received bevacizumab and CPT11. They did not report on their overall survival. So the jury is still out on overall survival with that trial. So that's the landscape as we sit and stand here today. We have some conflicting data from bevacizumab in the newly diagnosed glioblastoma setting. These, again, are fresh off the presses. We really have to have this data peer-reviewed. This data has to be published, and we have to really kind of sort this story out. It remains an approved drug as of today in the U.S., and I think, though, the jury is out on what the FDA will ultimately conclude about its full approval based on these data. But what is also clear is that we have to look at other proangiogenic signal transduction pathways, and I just wanted to leave you with a few comments on the post-VEGF world. This is just the cartoon I showed you earlier. There are many other important signal transduction pathways. There are other drugs that are targeting these pathways in phase 1-2 trials. Just to mention one of my favorites, which is angiopoietin. Angiopoietin signaling is quite complex, but the bottom line is that in the setting of VEGF suppression, of VEGF signaling, angiopoietin-2 is a proangiogenic factor, and it results in blood vessel growth, endothelial cell proliferation. Ang2 is overexpressed in 70% of the endothelium of glioma patients. We know that increased Ang2, Ang1 levels in all solid tumors, in multiple solid tumor models, leads to poor survival, increased metastasis, increased angiogenesis, and in gliomas, increased invasiveness. We've noticed in several of our studies with cediranib that Ang2 may be an important player. Here you can see that when we treat with an anti-VEGF therapy for recurrent GBM, what happens ultimately is Ang2 goes down but then comes up, and it correlates very nicely with radiographic progression in these patients. These are mouse models treated with various combinations of anti-VEGF, anti-Ang2 drugs. And the very best combinations that we've seen of any of these in the lab are the combined anti-VEGF, anti-Ang2 approach. So we have a number of molecules that are currently entering phase 1 and phase 2 trials built around the angiopoietin signal transduction pathway. And finally, c-Met. We know from models, animal models, we know from autopsy studies that patients treated with anti-VEGF agents upregulate c-Met signaling, and that c-Met expression can be on endothelial cells as well as on tumor cells. And this was an interesting case report in the Journal of Clinical Oncology. This was one of our patients who had failed anti-VEGF therapy, was growing, received crizotinib, which is a MET, as well as an ALK inhibitor. And this is one of the only cases I've ever seen after failure of an anti-VEGF therapy where there was a response, in this case about a 45% shrinkage if you actually measured out with crizotinib, which lasted about 6 months before the tumor progressed. So in summary, angiogenesis is critical. We all acknowledge that for glioblastoma growth. I've harped on it. There are multiple proangiogenic pathways. Unfortunately, as of today, no anti-angiogenic agent has yet to improve overall survival in a randomized phase 3 trial. There are multiple mechanisms of action which help us think about therapeutics. We must think about mechanism of action. And the treatment of our patients who fail anti-VEGF therapy is quite challenging, as these tumors may become more invasive and infiltrative, and that's why we have to focus on some of these pathways like Ang2 and c-Met. And with that, I'll thank you for your attention.
Red Lano, Red Latinoamericana de Neuro-Oncología.